In 1966, the wobble hypothesis proposed by molecular biologist Francis Crick suggested that the modified base at the first positions of the tRNA anticodon can pair with more bases of the mRNA codon than his standard theory of AU and GC base pairings. This rule is the beginning of the human exploration of diverse decoding mechanisms. More interestingly, three life domains of bacteria, archaea, and eukarya have diverse mechanisms for decoding the AUA codon for isoleucine. In eukarya, each yeast species could have different modified bases like inosine or cytidine in isoleucine tRNA, decoding for AUA codon in mRNA. Meanwhile, in bacteria, tRNA with cytidine conjugates to a lysine called lysidine, abbreviated as L, recognize adenine in AUA for isoleucine instead of guanine in AUG for methionine. Lastly, archaea have AUA codon deciphered by isoleucine tRNA with a modified cytosine, abbreviated as C+, asking for the similarity to lysidine in bacteria. However, no discovery of lysidine in archaea tRNA and homologs of tRNA isoleucine lysidine synthetase, abbreviated as TILS, in archaea genomes raise curiosity about the distinct chemical structure and its biogenesis of this archaea modified base. In 2010, an archaea haloacula maris mortuae in the eukaryotes film was experimented by Professor Yoshiko Ikeuchi et al. To isolate this cytidine modified base. Its unique molecular mass of 355 grams per mole is heavier than the normal cytidine. Mass spectrometry degraded the modified base into fragmented ions to uncover the conjugative molecules' fragmentation pattern and connectivity. After acceleration and detection, the respective recombination of fragmented ion interpret Agmatine was the only natural compound that satisfied this chemical structure, and no longer strange to us. Agmatine was synthesized from amino acid arginine through cellular metabolic pathways. Agmatine conjugates to oxygen at the C2 positions of cytosine through its primary amine group by deoxidation. The researchers suggested that the combination of agmatine and cytidine create the basic name Agmatidine for this archaea modified base, as the term lysidine was the fusion of lysine and cytidine in bacteria. But there are still a few questions in our mind. Is the mechanisms to create Agmatidine just simply like that? Do all archaea phyla have isoleucine tRNA for AUA codon with Agmatidine, and why not in bacteria? The answer is only three archaea phyla, Eukaryotes, Archaeota, and Archaeota, have the specific homologs in their genomes encoding for the enzyme tRNA isoleucine Agmatine cytidine synthetase, or we could call that briefly TIRS. To catalyze the Agmatidine formation, TIRS recognizes and binds to the anticodon arm of tRNA and then hydrolyzes ATP into AMP and pyrophosphate. Looking at the molecular structure of cytosine, TIRS uses the gamma phosphate from the pyrophosphate to activate the C2 position of cytosine as the phosphorylated intermediate and release the beta phosphate. Agmatine had already attached to TIRS before the enzyme bound to tRNA and then attacked the C2 position of intermediate through the amine group, leading to deoxidation and complete the Agmatidine formation. This modification causes tautomeric shifts to convert the amine group at the C4 positions of cytosine into the amino group and reduce a hydrogen bonding site, leading to the base pairings of Agmatidine with adenine instead of guanine. This archaea unique cytidine modification Agmatidine determines the codon specificity of isoleucine tRNA for AUA instead of AUG in the translation system. In the future, further insights will continue uncovering the functions of different TIRS motifs. for catalyzing Agmatidine formation in archaea isoleucine tRNA decoding for AUA codon, compared to bacterial isoleucine tRNA lysidine synthetase or TILS in catalytic formation of lysidine, and also how convergent evolution from different ancestor occurs in the decoding system for AUA codon. But that is the story of the future. See you soon.